Hi students, today let me talk about xenobiotics, detoxification or biotransformation. In our body we are having some foreign bodies like pesticides, cosmetics, drugs and food additives. They all produces some toxic chemicals in our body within 24 hours. Continuously they are producing. They call it as xenobiotics. Actually, these are all very toxic. So, this should be removed from our body. That is why the definition for xenobiotic is the toxic compounds are converted into non toxic compounds and then get excreted by kidneys. Okay, from our body, it should be removed. The site of this xenobiotics is liver because the enzyme is present in liver only, xenobiotic enzyme. What happens to this that is xenobiotic compounds, toxic compounds? Ultimately, they will go for cancer. That is why it should be removed. Okay. So the concept of this xenobiotic or biotransformation is the toxic compounds are converted into harmless or inactive compounds and then excreted by kidneys. So for this xenobiotics, we are having two phases of reactions, phase 1 and phase 2. In phase 1, the major reaction is hydroxylation. All toxic compounds should go for hydroxylation reactions. The concept is actually here, the mechanism is all these toxic are lyophilic compounds. These lyophilic compounds should be converted into hydrophilic compounds and then the excretion will be easy for the kidneys. Okay. So, in hydroxylation, we are having some examples like hydrolysis, oxidation, deamination, and reduction. The mnemonic is for me is HRD office, Human Resource Development, HRD office, HRDO, hydrolysis, reduction, deamination, and oxidation. For each one, you have to give some examples. In your book, actually, so many examples are there, but we expect one single example for the reaction. For example, hydrolysis. The example is aspirin is converted into salicylic acid by hydrolysis. Second one is oxidation. You all know ethanol is converted into acetic acid by oxidation. Then deamination, synthesis of benzoic acid from benzylamine is an example for deamination. Reduction is very simple. Picric acid is converted into picramic acid. Like that, for each and every reaction, you have to give one example. For all these reactions, we have only one enzyme for detoxification, that is cytochrome P450 enzyme. This is otherwise known as monooxygenase enzyme. So example for monooxygen means cytochrome P450 enzyme. You should know about the significances of or functions of the cytochrome P450. This is another separate as a short notes. I actually already uploaded one video for separate video for this cytochrome P450 where it is synthesized, what are all the functions like that. Okay, it is synthesized from liver. These are all about phase 1, that is hydroxylation. For that hydroxylation, the reaction is HRDO, hydrolysis, reduction, oxidation, and deamination. Okay, coming to the second phase, actually the importance of second phase is in phase 1, some compounds are not able to convert it into non-toxic compounds. That is why we will be going into another phase of reaction, that is called conjugation. Our aim is to excrete from our body the toxic compounds. So conjugation. Conjugation meaning is further modification for the excretion of the non-toxic compounds. Here the examples are ACSM. The mnemonic is ACM. Actually, ACS Medical College like that. Chennai we have ACS Medical College. So ACSM. That is acetylation, conjugation, sulphation, methylation. In this conjugation is very important reaction in phase 2. For this conjugation, you have to give an example for all these four, but especially conjugation you have to give important. For example, conjugation of the cyclophosphamide. This is one of the important anti-cancer drug by glutathione. This is the glutathione example. Second one is conjugation by glycine. You know all the products derived from glycine. Here the bile acid, that is cholic acid, is converted into glycocholic acid. This is the example for conjugation by glycine. Third one is conjugation by glucuronic acid. In the heme synthesis breakdown of heme, you know the catabolism of heme, the unconjugated bilirubin from the hemolysis is enters into the liver where it will converted into conjugated bilirubin. Here the enzyme is diglucuronil transferase. The compound name is glucuronate. So conjugation by glucuronic acid example is conversion of unconjugated bilirubin into conjugated bilirubin, and then this will be easily excreted by through bile and kidneys. Okay, so like that, conjugation also you have to give four examples. That is ACSM: acetylation, conjugation, sulphation, and methylation. For all, you have to give one example. And the next and last point is, what is the role of cytochrome P450? Why I am repeating this one is, this is very very important. So role of cytochrome P450, that is monooxygenase or otherwise heme containing enzyme, role is converts the xenobiotic compounds, that is pesticides, cosmetics, drugs and food additives, toxic compounds should be converted into non-toxic compounds by this cytochrome P450 enzymes. Then only it will get excreted. Okay, bye.